Okay, so guys, now let's continue with our discussion. Okay, so we have here now your endosomes. Okay, so your endosomes are examples of membrane-bound organelles. So just like the other organelles that we have discussed, okay, in the part one of this lecture, your endosomes, okay, are also membrane-bound. Okay, so your endosomes are actually just like your lysosomes, okay? So the endosomes are actually vacuoles that surround now the materials that were internalized, okay, during endocytosis. So kung maalala nyo yung process of endocytosis natin, the one no that surrounds now the endocytose particles is actually your endosome, or yeah, the your endosomes, okay? But of course, sabi ko nga parang kamukha siya 'di ba nung lysosomes nyo, because your lysosomes are actually vacuoles then, okay? It's just that the difference, okay, of your endosomes from that of your lysosomes is that your lysosomes contain now hydrolytic enzymes. So if you still remember in phagocytosis, okay, the engulfed microorganism, for example, okay, in the process of phagocytosis, are actually digested, okay, by the cell, okay, through its lysosome. So, yung engulfed na na microorganism natin, 'di ba? It will be surrounded in a vacuole, okay? It is actually found inside your phagosome. Okay? So, yung phagosome na yon will actually fuse with your lysosome, and the fusion of your phagosome with your lysosome will create now your phagolysosome. Okay, and the hydrolytic enzymes, okay, in your phagolysosome will actually cause now the digestion of these phagocytosed microorganism. Okay, so guys, no, sorry, medyo um, maano yung pagsasalita ko kasi I just had my extraction, tooth extraction, and I'm so worried kasi na-dislodge ko yung clot niya. So, medyo may pain. Okay? So, please, no, bear with me na lang. Okay? So, guys, no, your endosomes, just like din nung ating Golgi apparatus, okay, or the Golgi bodies, it's also a sorting organelle. Okay? So, it regulate now the traffic of proteins inside the cell. And when we say protein, we're referring to those that are being endocytosed by the cell. Okay? So before they are being delivered, okay, dun sa kanilang area or the organelles to where they should be, okay? They are actually carried by or are surrounded by your endosomes. So again, your endosomes are much like that of your lysosomes. The only difference is that they are also vacuoles, but unlike your lysosomes, they do not contain now your hydrolytic enzymes. Okay? So for your endosomes, no, we can classify them as either your early or your late endosomes. Okay? So your early endosomes are membrane enclosed compartment, okay, associated with all the endocytic pathways. So what are the different endocytic pathways that we have discussed last time? We have your pinocytotic pathway, we have your phagocytosis, and your receptor-mediated endocytosis. So, sabi niya dyan, they are associated, okay, with all these endocytic pathways. Okay, so they're restricted to a portion of the cytoplasm near the cell membrane. So that's your early endosome. So based on this description, we can see it, or we can see here that if this is the nuclear membrane, sorry, this is the plasma membrane or the cell membrane, yung mga early endosomes natin, guys, dito sila, okay, near the periphery. Medyo gilid sila na portion. Okay? So, yung mga endocytose particles, proteins like that, they are actually carried by these endosomes na malapit sa mismong cytoplasmic membrane or your plasma membrane or the cell membrane. Okay? So, they are your early endosomes. Okay? And then we also have your late endosomes. So they are vesicles originating in early endosomes and traveling to the deeper, okay, structures in the cytoplasm. So these are the ones, okay, that are actually near no dun sa mga cytoplasmic organelles natin kung saan no dapat mapunta yung mga endocytic particles natin. Okay? So these endosomes, okay, can actually develop, or your early endosomes I should say, can actually develop into your late endosomes. So mamaya makikita natin, ma'am, so ibig po bang sabihin your early endosomes are actually or your late endosomes I should say are actually the mature early endosomes. Okay, so we could have kasi different models, okay, on how these two are actually formed. Okay, so mamaya pag-uusapan natin sila. Okay? So, again, ang tatandaan ninyo, your early endosomes, they are usually confined dun sa areas near your plasma membrane. Okay, so they are usually found at the periphery of the cell, and that of your late endosomes naman are usually, okay, found doon sa ating central portion ng cell, and sometimes or deeper portion, okay, nung ating cell. And sometimes, okay, they are actually now your lysosomes. Okay, they mature as your lysosomes. So your early endosomes to your late endosomes then eventually become your lysosomes. Okay, so hold on. Okay. So guys, now we have here the characteristics of your early endosomes and your late endosomes. So again, for the cellular localization, when we say cellular localization, we're talking about no in what part of the cell, okay, are these endosomes being found, or in what part of the cell are these endosomes located? So for your early endosomes, they are found in in the more peripheral cytoplasm. So again, malapit po dun sa ating plasma membrane. Okay? And then for the morphology, they have usually tubulovesicular structure, and their state of acidification, they exhibit only a slightly acidic environment. Okay, that is usually at 6.2 to 6.5. Bakit kailangan nyo tong tandaan, guys? Because the deeper the endosomes, no, doon sa ating cell, the more acidic they are. Okay? And eventually, if they mature as to become your lysosomes, mas acidic sila. Okay? And it's actually a requirement for these vacuoles to be able not to digest now the endocytose particles. Okay? So function again is to sort and recycle your protein internalized by endocytotic pathway. So tatandaan nyo to ha, recycle protein. Guys, do you remember doon sa ating receptor-mediated endocytosis, di ba meron tayo nung clatrine? Other than the clatrine, we have there your adaptin, di ba? So sinabi ko sa inyo, after na 'di ba, they are used 'di ba to coat now yung ating mga vesicles, but after na na-endocytose yung ating cargo protein 'di ba na-uncoat yung ating vesicle. Okay, the uncoating will actually um cause now the removal of your adaptin and your clathrin surrounding the vacuole. Okay, and these removed adaptin and your clathrin are actually recycled. Okay, so they are actually delivered back into your your early endosomes for recycling. Okay, para magamit dun sa next endocytic pathway natin. Okay. Okay. So next, we also have there your late endosome. So guys, no, if you can see yung ating late endosome, they are usually positioned near the Golgi apparatus and the nucleus. Okay, sa Golgi apparatus po natin and that of your nucleus. Okay? So adding, ibig sabihin, nasa deeper portion siya. If this is, for example, no, your cell, we have here your cell membrane or your plasma membrane. And then we have here your early endosomes, okay? And then we have here your nucleus, okay? And then the rough ER, yeah, tama ba ako? The rough ER, okay? And then we have there the Golgi apparatus, okay? So nearer here in this portion, okay? So andyan po yung ating um I'll call this one your late endosomes. So kung mapapansin ninyo, mas malapit sa gitna or deeper area yung ating late endosomes compared to your early endosomes nandito sa malapit sa inyong plasma membrane. Okay? So guys, no, their morphology, they have onion-like internal membranes. Okay? And kung mapapansin po natin, yung state of acidification niya, it's it's more acidic. Okay, so it averages to 5.5 pH of 5.5. Unlike po yung early endosomes ninyo, it's slightly or it's less acidic. Okay, so that's for your late endosomes. Okay, so guys, now we have here no a picture or a diagram. Hold on. Showing to us now, okay, the fate of the protein, okay, that are being endocytosed doon sa ating cell or sa ating plasma membrane, okay? So if you can see here, what type of ano to, endocytosis? Adding this is actually your example, your receptor, di ba? Mediated endocytosis. So if you can see here, we have here your clathrin that helps now form the pit and eventually your coated vesicle. Okay? So receptor mediated, okay, endocytosis. So guys, now if you can see, we have here your coated vesicles, okay? So that eventually will deliver now the endocytose, okay, cargo protein together with the other proteins like your receptor protein, your how you call this one? Your coated your clathrin that coats the vesicle. Okay, so this is your early endosome. Ito po siya. Okay, your early endosome that is at pH of 6.2. Okay, so again, di ba, other than it sorting the protein, okay, it also actually recycle now the proteins that were used during the endocytosis. So kung makipapansin po ninyo, itong mga receptor proteins natin, they are being recycled by the early endosome to be delivered back, okay, doon sa ating surface, sa may plasma membrane po natin. Okay, for another endocytotic pathway. Okay? And then, of course, after, no, nai-sort po niya yung ating mga proteins. Okay, kung mapapansin nyo dito na sort po niya siya. Okay, these endocytose protein now or proteins are being transported in your MVB or also known as your multivesicular bodies. So these multivesicular bodies will later, okay, transport these proteins, okay, in your late endosome. So this is an example of your late endosome. So they are found deeper, okay, doon sa ating cell. And the pH is usually at pH 5.5. So it's more acidic compared to the pH of the environment in your early endosome. Okay? And then from your late endosome, okay, which is usually found no, near the Golgi apparatus and the nucleus, the protein is transported now in the lysosome. Okay, so proteins are being transported into the lysosome where the pH, of course, it's much more acidic, okay, compared to that of your late and your early endosome. So the pH is at 4.7. So this is where the proteins, okay, are being degraded. Okay, kasi nga sabi natin, if we're going to compare your lysosome from that of your endosomes, they are actually vacuole surrounding now yung endocytose material. It's just that the difference is that yung lysosome po natin ay meron po siyang mga hydrolytic enzymes. Okay? So your endosomes, okay, can be viewed as either stable cytoplasmic organelles or as transient structures. Okay? So we have two different models, guys, that explain now the origin and formation of your endosomal compartment. So we have two models here. We have your stable compartment model, okay, which will describes now that your endosomes are actually stable organelles. So meron daw tayong separate na early at late endosomes. So they are separate organelles and they are also separate organelles from that of your lysosome. Okay? And then we also have your maturation model. Okay, sa maturation model naman ating, sabi niya, si early endosome po natin ay nafo-form and then it becomes now or it matures to become your late endosomes and then your late endosomes mature now to become your lysosomes. So, unlike your stable compartment model, for the maturation model, sabi niya, yung mga endosomes natin, they will eventually become now your lysosomes. Sa stable compartment, ang sabi po niya, they are actually separate organelles. Your early endosomes are different from that your late endosomes and from that of your lysosomes. But in your maturation model, it says that your early endosomes will mature to become your late endosomes and eventually will also mature to become your lysosomes. Okay, so these are the models that explain the origin and formation, okay, of the endosomal compartments. Okay, so this time, hold on. Okay, so next is lysosomes. Okay, so for your lysosomes, okay, we'll be talking about the formation, okay, of our lysosomes and the pathway, okay, involved now in the formation or in the delivery, I should say, of endocytose microorganisms or micro particles, I should say, okay, in the lysosomes. Okay, so guys, now we have here your lysosome biogenesis. So this diagram shows now your regulated and constitutive pathway for the delivery of lysosomal specific membrane proteins in early and late endosomes. Okay, so these lysosome specific proteins are synthesized, okay, in your endoplasmic reticulum, and then they are transported to your Golgi apparatus and reach their destination by two pathway. Okay? So, ang tinutukoy po natin dito, guys, no, is yung formation nung ating um, lysosome, okay, by delivering now the lysosomal specific membrane proteins coming from that of your early and your late endosomes. Okay, so these proteins are synthesized, okay, in your rough endoplasmic reticulum and of course are sorted and packaged and transported from your Golgi apparatus and eventually to its destination, okay, with the pathway or through pathways, okay, which is either your constitutive secretory pathway. Malala nyo yung sa exocytosis natin, di ba? We have there your constitutive and regulated exocytosis. Dito naman sa lysosome biogenesis, meron tayong constitutive secretory pathway and your endosomal Golgi-derived coated vesicle secretory pathway. Okay? So guys, no, kung mapapansin ninyo, this is your constitutive pathway, itong pong blue. Okay, so this is no your Golgi apparatus. So if you can see here, we have the specific lysosomal proteins. Okay, so in the constitutive secretory pathway adding, this is a continuous process, just like how we have defined um, your pinocytosis, which is an example of a constitutive pathway, together with your constitutive exocytosis. Okay. So guys, no, in your constitutive secretory pathway, your specific lysosomal proteins, okay, will exit now your Golgi apparatus. So this one, it will exit it and they will be actually delivered, okay, dun po sa surface ng ating plasma membrane. So this is the membrane and your lysosomal proteins are delivered there, okay? And then these lysosomal proteins are actually endocytosed. Okay? So they will be endocytosed. That is by the what? By the formation now of your clathrin-coated vesicle. Napansin nyo, ito siya. Okay? And then eventually, this clathrin-coated vesicle that contains now your lysosomal or specific lysosomal proteins will be delivered to your early endosome. Okay? And then your early endosome, or from your early endosome to your late endosomes, and eventually to your lysosome. So again, adding, di ba, remember in the formation no, of your um, early and late endosomes, so we have the stable compartment model or the maturation model. So it's either your early endosome maturing to become your late endosome, or early endosome as separate organelle from your late endosome. Okay, so this is your constitutive pathway. Okay, so again, your specific lysosomal proteins are delivered in the surface of the membrane. They are being endocytosed, okay, via your clathrin-coated vesicles, and eventually are delivered to your early endosomes, then to your late endosomes, and finally to your lysosomes. Okay, so they will make now no yung ating lysosomal membrane. Okay. And then guys, no, for the Golgi-derived coated vesicle path or secretory pathway naman, ito pong green natin, okay? So sabi po niya dyan, after packaging and sorting of these proteins doon sa ating Golgi apparatus, the Golgi or the specific lysosomal proteins will be pinched off, okay? From your Golgi. So napansin nyo, dun sa constitutive pathway, they still have to reach no, the plasma membrane to form a vesicle. But in your Golgi-derived coated vesicle secretory pathway, yung mga specific lysosomal proteins natin, they form po, um, sorry, they form now po yung ating vesicle from the Golgi mismo. Okay? From the membrane of the Golgi. Okay? Kaya siya tinawag na Golgi-derived. Okay? So, ganun din, they will be pinched off from the Golgi membrane. So, there will be a formation of a vesicle that's, again, latrine coated. Okay. Then after that, these okay vesicles will be delivered to your early endosomes and then to your late endosomes. Okay. So they are delivered both in early and late endosomes and eventually to your lysosome. So kung mapapansin niyo guys, no, yung Golgi derived natin, they are being delivered to early and late. So parang ito po yung ating how do you call this one? Um stable compartment model. Meron tayong early, meron laga pa tayong late. Okay? While in your constitutive secretory pathway, it has to, the protein or the coated vesicle has to reach first the early endosome, then eventually your late. So maybe this early endosome becomes now your late and eventually becomes your lysosome. So ang minomodel naman niya, maring ito yung pathway na nangyayari dun sa ating maturation model. Okay? So, kapag ka naman yung stable compartment, pwedeng yun sa Golgi derived. Okay? But of course, studies should be conducted to confirm this. Okay? So, but if you can see or try to analyze this diagram kasi, in your Golgi derived, the proteins are being delivered both in your early and your late. Okay, so ibig sabihin, may separate talagang early endosome at late endosome. Okay, that can be proven, okay, or that can be, how you call this one? This is an indication, okay, of the stable compartment model. Okay, but in your constitutive secretory pathway, maaring stable compartment model to, o pwede ding baka yung early endosome natin nagmamature to become your late and then your lysosome. Okay? So guys, no, your lysosomes, they are spherical with a diameter of about 0.5 to sorry, 0.05 to zero point 0.5 micrometer in diameter. So they present a uniformly granular electron-dense appearance, okay, under your transmission electron microscope. Okay? So guys, no, kung mapapansin po natin yung loob niya, meron sabi niya dyan, uniformly granular electron dense. Okay? Those are actually containing now yung mga dense particles, okay, na nakikita po natin dito sa loob niya. is actually yung mga hydrolytic enzyme, or yung mga are actually your hydrolytic enzymes. Okay, so if you can see here, na-appreciate din po natin yung kanyang membrane. This is the blow up image of this membrane. Okay, it has also the lipid bilayer. Okay. So your membrane limited vesicles, or it's a membrane limited vesicle, okay, and that they contain about 40 hydrolytic enzymes. So tandaan po natin guys, the lysosomal enzymes are actually synthesized in your rough endoplasmic reticulum, and these are being sorted, okay, in the Golgi apparatus based on their binding ability to the M6P receptors. Okay, so the most common hydrolases that we can find no, in your lysosomes are the following. We have your proteases, your nucleases, phosphatases, phospholipases, sulfatases, and your beta-glucuronidase. Okay? So lysosomes, unlike other membrane-bound organelles, okay, have now unique membrane. That bakit unique po yung kanilang membrane? That what's make it resistant po dun sa mga hydrolytic enzymes na nasa loob niya. That's why the hydrolytic enzymes, of course, are being contained sa lysosome at hindi po nadadamage yung membrane niya kasi kakaiba po yung structure niya compared to the membranes of the other organelles. Okay? So the lysosomal membrane has unusual phospholipid structure that contains cholesterol and a unique lipid called lysobiphosphatidec acid. Okay, and they also contain now your membrane proteins. We have your LAMPs, or your lysosome associated membrane proteins, your lysosomal membrane glycoproteins, or your LGPS, and your lysosomal integral membrane proteins, or your LIMPs. Okay, so this is the structure, or this is how, okay, your lysosomal membrane appear. Okay, so if you can see here po, mga adding, okay, we have these proteins, okay, that are being synthesized, okay, in the rough endoplasmic reticulums, and they have specific lysosomal targeting, okay, signal. So we have here the membrane. So this is a membrane that is impermeable to enzymes. Okay, and that what makes it not unique from the membranes of other membrane-bound organelles. Okay, so your lysosomes and late endosomes contains proton pumps and transport proteins. Okay, that transport now final product for digestion. So guys, kung mapapansin niyo, itong mga, 'di ba, dito po yung ating membrane, okay? So these are actually your membrane proteins, okay? So they serve as your transport proteins. So we have here your proton pump, okay? Your hydrogen pumps, okay? We also have here transport proteins, okay? Or pumps, okay, for other particles, okay? Like your organic link phosphates, sulfates, transport protein for your nucleic acid, for other proteins, for your polysaccharides. Okay, and inside are your enzymes. Okay, so if you can see, if we are going to transport now here, yung nucleic acid, they are being digested or hydrolyzed, okay, by your nucleases and other related enzymes. Okay, for your organic link sulfates, they are being degraded, okay, by your sulfatases, okay, and so on. Okay, so this is your lysosome, and of course the associated membrane proteins. Okay, so guys, now we have here the clinical significance of your lysosome. If you have heard already of chloroquine, chloroquine is an example of a drug that we use for the prevention of malaria. Okay, so it's a lysosomotropic agent, okay, that accumulates in the lysosomes. Diba kanina guys, we have compared no yung pH po nung ating, how we call this one, the late endosome, the early endosome. And if you can see there, your early endosome has an acidic pH, your late endosome has more acidic pH, and eventually your lysosome has, okay, much, much more acidic high pH. Okay, so guys, no, what is okay the sorry sakit ano. So what is okay the how do you call this one? The mechanism, okay, involved in the use of your chloroquine for malaria or as an anti-malarial drug. Okay, guys, no, your chloroquine actually has the ability to raise, okay, the pH of your lysosomal content. Di ba acidic po sila? Okay, but your chloroquine has the ability to raise the pH of the lysosomal content, and raising the pH now, okay, or making it alkaline or less acidic, okay, would actually render, okay, the lysosomal enzymes inactive. Okay, ibig sabihin they will not they're not able to do no yung kanilang function because of the unfavorable environment. Kasi nga, di ba, they require an acidic pH that is about a pH 4.7. But if you're going to raise the pH higher than 4.7 to make it a little more alkaline, okay, or less acidic, that is actually not favorable, okay, for your lysosomal enzymes, okay, for them to be activated and perform, okay, their function of digestion. Okay? So, yun po yung mechanism sa chloroquine natin. So, ano nangyayari? Since there's rising, okay, pH in the lysosomal content, nagkakaroon tayo ng inactivation ng lysosomal enzymes po natin. Okay? So, that accounts now to the anti-malarial activity, okay, of your chloroquine. So, anong nangyayari? The drug concentrates in the acidic food vacuole of the malarial parasite. Okay, example, your Plasmodium falciparum. Okay, and then this interferes now with digestive process. Okay, so ibig sabihin adding, how will, okay, the falciparum, okay, be able to benefit from the food, okay, that it's actually needing, no, from its food vacuole. Kung hindi po siya nadadigest, okay, nagkaroon tayo ng disruption ng digestion. Why? Because, no, of your chloroquine raising now the pH, sorry, to a less acidic one. Okay? So that eventually will lead now to the killing of your parasite. Okay? So the interference, okay, in the digestive process will starve now your parasite and eventually kill it. Okay? So that's your chloroquine. So ulitin ko, what's the action, okay, of your chloroquine? It raises, okay, the pH of your lysosomal content, thus making now, okay, the digestive process in the um, a parasite or malarial parasite being interfered. Okay, so nagkakaroon tayo ng inactivation ng lysosomal enzymes and eventually will lead to the killing, okay, of the microorganism due to an interference in their digestive process. Okay, so there are three pathways of material delivery for intracellular digestion in lysosomes. So adding meron po tayong tatlo. We have the extracellular large particle, extracellular small particle, and the intracellular particles. Okay? So guys, no, for the extracellular large particles, sabi niya po dyan, we have bacteria, cell debris, and other foreign materials. Okay? So these foreign materials are being engulfed, okay, by the process of phagocytosis. Tandaan ninyo yan na extra large. Okay? So adding, we're referring to what? These are actually processes of your endocytosis. Okay? So for extracellular small particles, mapapansin natin guys, we have there, no, your extracellular proteins, plasma membrane proteins, and your ligand receptor complexes. Example, we have your endocytotic pathways such as your pinocytosis and your receptor-mediated endocytosis. So they are under po your extracellular small particle. Okay? And then for intracellular particles, so when we say intracellular, they are okay within your cell. Okay? We have your entire organelles. The organelles that have already served their purpose, ganito po sila na deliver sa ating lysosome. Okay? Your cytoplasmic proteins and other cellular components. Okay? So these components, guys, no, are being isolated, no, by your endoplasmic reticulum membrane. Okay? And then eventually are transported to your lysosome and degraded by the process we call as your autophagy or cell eating. Okay? So adding tandaan ninyo, I think lady. So guys, tandaan ninyo, no, for material delivery, we have three processes. We have extracellular large particles and the extracellular small. For the extracellular large, we're actually referring, okay, dun po sa ating phagocytosis. And for the extracellular small, we're referring po doon sa ating pinocytosis and your receptor-mediated endocytosis. Tandaan nyo kasi, we're discussing about delivery. These are your endocytotic pathways, okay? And finally, we have intracellular particles. So we're referring to organelles, cytoplasmic proteins, okay, that are being degraded in your lysosomes and are delivered, okay, in your lysosomes through the process of your autophagy. Okay? So, we have here the pathways of delivery of material. Ulitin ko na naman. Ito na naman sila, guys. The endocytic pathway, the phagocytotic pathway, and the autophagic pathway. Malala nyo, yung endocytic pathway, ano to? These are your extracellular small particles. Under these are your pinocytosis, receptor-mediated endocytosis. Okay? Pag phagocytotic, ano to? These are your extracellular large particles. What are these? These are your bacteria, your cell debris, and other foreign materials. Okay, and then finally, we have your autophagic pathway. Okay, so for autophagic pathway, we're talking about cell organelles. Okay, what else? We have cytoplasmic proteins, okay, that are being engulfed or being covered by the endoplasmic reticulum membrane and eventually are actually degraded in your lysosomes, okay? So we have here a diagram showing to us, no, the pathways of delivery of materials for digestion in your lysosome. So guys, no, mas tatandaan ninyo itong tatlo po na ito. These are actually the general pathways of endocytosis. We have number one for extracellular large particles, just like your cell debris. We have your bacteria and of course other foreign materials. We have through the process of phagocytosis. Okay, natatandaan nyo yan sa endocytosis natin. And then, for your extracellular small particles, we have for example yung mga um, water molecules natin. Okay, what else? We have your proteins, okay, ligand receptor complexes. Malala niyo yan, yung sa receptor-mediated protein, ligand receptor, example, yung cargo protein cargo receptor complex. Okay, if you still remember that. Ito po sila. Okay, extracellular small particles. We have pathway of delivery, receptor-mediated endocytosis, and pinocytosis. Okay, so kung mapapansin nyo guys, ito yung diniscuss natin last time, di ba? Sa endocytosis, we have your phagocytosis, your pino, and receptor-mediated endocytosis. Okay, so guys, now we also have here the last one is your autophagy. Okay, so for the autophagic pathway, if you can see, we're referring, okay, to particles like, for example, the cell organelles that have already served their purpose. Okay, so they're being, how do you call this one, coated, okay, or isolated by a membrane. Like, for example, I should say, is the endoplasmic reticulum membrane. Okay, so this membrane surrounds now the organelles. Okay, we have here an example, your mitochondrion. So your isolation membrane, which is actually your SCR membrane, will isolate now this mitochondrion to form your autophagosome and deliver this one po doon sa ating lysosome for digestion. Okay? So guys, naintindihan nyo ba or nako-connect nyo ba yung napag-aralan natin last time? So kung maalala nyo, ulitin ko, when we say pathways of delivery, we're actually referring, okay, to your endocytotic pathways plus your autophagic pathway lang. Okay? So, ulitin ko. Pagka phagocytosis yan, extracellular, large particles. Your bacteria, your cell debris, and other foreign material. Pagka receptor-mediated endocytosis, pinocytosis, we have your what? Extracellular, small particles. Halimbawa, yung extracellular proteins natin, plasma membrane proteins, ligand receptors. Kung maalala nyo sa receptor-mediated endocytosis, di ba? We have there, guys, yung inyong, how you call this one? We have there your cargo, protein cargo receptor. Okay? Complex. So, yan siya. Okay, sila to. Okay? And finally, for autophagic pathway, we have your intracellular particles. So, when we say intracellular particles, again, we're referring to, the how you call this one? Sorry for that, no. We're referring, okay, to entire organelles, cytoplasmic proteins, and other cellular components, okay, that are being isolated from your um, endoplasmic or cytoplasmic membrane or matrix, I should say, by the membrane of your smooth endoplasmic reticulum. Okay, so those are the different pathways of delivery, okay, of materials for digestion in your lysosome. Okay? So guys, now let's move doon po sa ating next. Okay? So, okay. So these are the three well-characterized pathways of autophagy. So ito din po kanina yon. Okay, we have discussed no about autophagy kanina. Okay, so we have three. Okay, we have your macroautophagy, your microautophagy, and your chaperone-mediated autophagy. So ma'am, ano na naman ito? Okay, so guys, ano lang to, parang classification lang nitong autophagic pathway. Okay? So parang yung kanina, last time, 'di ba, sa endocytotic pathway nyo, meron tayo nung receptor-mediated pinocytosis. Okay? Actually, your phagocytotic is under there also. Okay? Ganun din dito sa ano nyo, sa autophagic pathway, meron po tayo nung tatlo. We have three, okay, subclassification or subtypes. We have your macroautophagy, microautophagy, and chaperone-mediated autophagy. So, ading depende lang naman ito dun sa kalaki, okay, nung material that is being, how do you call this one, delivered, okay, via autophagy. Okay? So, let's look into it. Okay? Wait lang nahirapan talaga ako sa pagsasalita. Okay? Okay, so first we have your macroautophagy. Your macroautophagy is a non-specific process, okay? So here, a portion of the cytoplasm or an entire organelle is first surrounded by the isolation membrane. Ito yung example niya, Adi, no? This one is your macroautophagy, okay? So your isolation membrane is actually referring to a portion of your smooth endoplasmic reticulum membrane. So this one will isolate now the organelle, okay, to be delivered via autophagic pathway. Okay? So na-surround niya. Ito na siya. That's to form now your autophagosome. So adding, that's your macroautophagy. So we're referring to the isolation, okay, of an entire organelle or a portion of your cytoplasm. Okay, and that's to form now your autophagosome, okay, which matures into your lysosome. Okay, so isolation membrane disintegrates within the hydrolytic compartment of a lysosome to expose now the organelle, okay, that has been delivered via autophagic pathway. Okay, so ulitin ko, anong klase ng autophagy to? Ito po ay macro-autophagy because we are actually referring to the autophagy of a cell organelle or a portion of the cytoplasmic matrix. Okay? So next, we also have your micro-autophagy. Okay? Your micro-autophagy is also a non-specific process. Okay? So dito naman, micro, ibig sabihin mas maliit po siya. Unlike in your macroautophagy na ang tinutukoy po natin ay cell organelle, dito sa microautophagy, we're referring to cytoplasmic proteins. Okay? So medyo mas maliliit po sila. And they are actually degraded in a continuous process. So this is also a form of a not of a constitutive pathway. Dere-derecho. Okay? So small cytoplasmic soluble proteins are internalized by invagination of the lysosomal membrane. So nagkakaroon po tayo ng invagination sa lysosomal membrane and they will actually internalize now, okay? Or parang kumbaga niyayakap or kinukulong nila yung mga soluble proteins for it or for these proteins to be internalized sa loob ng ating lysosome for degradation. And that's your microautophagy, okay? And then another, okay, pathway, okay, of autophagy is what we know as your chaperone-mediated autophagy. So unlike no yung ating microautophagy and macro, which are non-specific processes, your chaperone-mediated autophagy is a selective process, okay, of protein degradation. So it requires now, okay, certain chaperone or a protein that will tag, okay, yung ating protein for lysosomal degradation. So, yung protein po na kailangan nating i-degrade, kailangan munang samahan nung isang protein na to. And that's yung heat shock chaperone protein or also known as your HSC73. Okay? So guys, no, this process is usually activated during nutrient deprivation and also responsible for about 30%, okay, of cytoplasmic proteins in organs such as your liver and the kidney. So yung autophagic pathway, okay, that usually happen, okay, dun sa ating liver and kidney, okay, is of this type. We have the chaperone-mediated autophagy. Okay? So, ito po yung ating diagram or yung picture showing you now no, how these pathways, okay, are actually being than, okay, by the body, okay? So we have your macroautophagy. Ito po siya yung ating organelle, entire organelle. So ang una is the formation of your autophagosome, 'di ba? The autophagosome is formed now by how you call this one? By the isolation of this organelle, okay? By your isolation membrane or yung membrane po ng ating endoplasmic reticulum, your smooth endoplasmic reticulum. So, surround niya po yung ating organelle and that forms now your autophagosome. And eventually, okay, it will mature or it will become your lysosome or it will fuse. In some books kasi it says there that it will become your lysosome, okay? Or in some books, or just like here now, it says it fused, okay, with your lysosome. Okay? And then from there, it's degraded. Okay? And then for your microautophagy naman, parang ganun din siya. Macroautophagy is just that there's no need for the isolation membrane. Yung maliliit na particles lang natin, automatic lang sila pagdating po nila dito sa membrane nung ating lysosome, nagfo-form po ng invagination. Nakikita nyo to, parang mayroong mga, how you call this one, parang lumalalim yung plasma membrane. That is to, this is an invagination. Okay? 'Di ba? So that is to, how you call this one, to internalize, okay, yung mga proteins na to, cytoplasmic proteins na to, papunta sa lysosome natin, okay? And of course, for digestion, okay? For chaperone-mediated autophagy, kamukha rin siya guys, no, nung microautophagy nyo. It's just that in your chaperone-mediated, there's a need for your HSC73 or your heat shock protein 73, okay? So this protein will bind now the protein 4 lysosomal digestion. Okay, so this will help the protein to be transported into the lysosomal lumen, okay, for degradation. Okay, so again, those are the different pathways of autophagy. We have your macroautophagy for your cell organelles, your microautophagy for your cytoplasmic proteins, and of course your chaperone mediated autophagy that requires now the transport, okay, of certain proteins, okay, with the help of your chaperone mediated, or sorry, with the help of your heat shock, okay, protein 73, okay. So guys, now we have here an electron microgram of your autophagosomes in your hepatocyte, okay, in your hepatocyte, okay. So if you can see here, we have here the transmission electron image, okay. So kung makikita po ninyo dito, we can see here, no, several autophagosomes containing degenerating mitochondria. So these are actually your mitochondria. They were already introduced dito sa loob po ng ating lysosome and are being degraded. So kung makikita nyo, faint-like na lang po sila. Okay? So they are faint-like, okay, organelles dito sa ating transmission electron. Okay? So next, we have your heterolysosomes. So guys, now your heterolysosomes are active organelles, okay, and are generally larger, okay, and have more heterogeneous appearance in your transmission electron. Why? Because of the variety of materials, okay, that they may be digesting. They are actually your lysosomes then, but they are the ones that are active, okay, in doing their function of digestion. Okay? So, mas malaki sila kasi they contain now, okay, madaming materials na dinadigest nila. And heterogeneous sila, okay, in appearance under your transmission electron microscope. So, this one is an example, okay, of your heterolysosome. This one here. These are your lysosomes, okay? So guys, no, we have your residual body. Okay, what are your residual bodies? So guys, no, your residual body are actually resulting, okay, from the hydrolytic breakdown, okay, of the contents of your lysosome. So kaya nga sinawag silang residual body. Okay, so they are actually debris-filled vacuole, okay, that results from the breakdown of the contents of your lysosome, and they are, okay, normal features of cell aging. Okay? So in neurons, for example, okay, that underwent, no, or in aging neurons, for example, we have, no, yung accumulation nung ating, or these residual bodies, I should say, are accumulating as your lipofuscin. Okay? So, lipofuscin. So, you will know more of this pagdating nyo sa histopath and in other subjects like in clinical chemistry, okay? And I think even in hematology, you can encounter this one. Okay? So, ulitin ko, residual bodies, okay? They can accumulate as lipofuscin and you usually observe this, no, sa mga aging neurons natin. Okay? So guys, now we also have here the clinical significance of your lysosomes. We have the lysosomal storage diseases. Okay? So your lysosomal storage diseases are caused by pathologic accumulation of undigested substrate in residual bodies, okay, due to absence of certain lysosomal enzymes. So, yung mga residual bodies natin, guys, meron daw silang mga undigested substrate. Okay? So, ano nangyari? Bakit nagkaroon ng mga undigested particles? This is due to the absence of protein or of the enzymes necessary for the degradation, okay, of these particles. Okay? So these are the problems, okay, that we can observe in patients, no, with lysosomal storage diseases. Okay? So guys, no, please memorize this one. You will encounter this pagdating po ninyo sa clinical chemistry at kailangan kabisado po ninyo ito. Okay? So we have here the summary of common, okay, lysosomal storage diseases. Anong kakabisaduhin po natin? Yung kanilang pangalan, we have your Gaucher's disease. What's the deficient enzyme? That's your glucocerebrosidase. At ano yung nag-accumulate na product? Okay, it's your glucosylceramide. Okay? We also have your Tay-Sachs disease. The missing enzyme is your beta-hexazominidase alpha subunit, okay? And the accumulating product is your GM2 ganglioside. Okay? So please try to memorize yung pinaka-common po natin. Okay? Ito yung mga madalas dyan eh. Disorders of sphingolipid degradation. Ito. Gaucher's, Tay-Sachs, Sandoff, Scrub, Niemann-Pick. You will encounter this pagdating nyo sa CC. So please do memorize it. Okay? And then we also have your um, LSDs, okay? Disorders of glycosaminoglycans degradation naman. We have your Hurler syndromes, your Hunter, okay? We also have your single enzyme deficiency, your Pompe, or also known as your glycogenosis 2. Ang wala po sa kanya is alpha-1 for glucosidase. The accumulating product is glycogen, okay? So guys, please memorize, okay, all of this, okay? So, yan. So, those are the different, okay, LSDs or lysosomal storage diseases. Ano nga ulit ang problema? Nagkaroon po tayo ng mga undigested, okay, particles dun sa ating residual bodies due to the absence, okay, of proteins or enzymes in our lysosome. Okay? So, ito po sila pakikabisado. Okay? So, okay. So this time, let's move on to the proteasome-mediated degradation. So your proteasome-mediated degradation is used, okay, by the cell to destroy abnormal proteins, okay, that are being misfolded, denatured, or contain now abnormal amino acid, okay? So it degrades normal short-lived regulatory proteins that need to be rapidly inactivated. Okay? Example are your mitotic cyclins, okay, those that regulate cell cycle progression, transcriptional factors, tumor suppressors, or tumor promoters. So, kailangan sila kaagad sirahin, di ba? Yung mga proteins na to, okay? Otherwise, they can lead to what? Okay, for example, if hindi natin nasira kagad yung tumor promoters, they may lead to what? How do you call this one? Formation of tumor, okay? Progression of cancer, something like that, okay? So they have to be are rapidly inactivated. Okay? So these proteins, guys, no, are being okay degraded through the function or through your proteasomes, okay, or through the proteasome mediated degradation. Okay, so guys, now we have here the proteasome mediated degradation. Okay, so this shows now to you yung pathway kung paano po nangyayari yung degradation nung mga proteins natin, okay, sa inyong proteasome complex. Okay, so guys, now this is, okay, una, let's discuss first the proteasome complex. This is your proteasome complex. Etong buo na to, okay? So, guys, no, yung proteasomes complex po natin, it has, okay, the following components. We have your 19S-RP protein or regulatory protein. We have your 20S core protein or 20SCP, okay? And your 19SRP, okay, or regulatory protein. This one, 19SRP, is the 19SRP in the lead. Ito yung pasukan, okay, ng protein natin. And this one naman is the base. Okay, 19 SRP at the base. And this one here is the 20 SCP or core protein. Okay, the combination of these three, okay, is known as your 26S proteasome complex. Okay, so when we say when we're referring to the 26S proteasome complex, we mean your 19SRP, 20SCP, and your 19SRP at the base. Okay. So guys, paano ba nangyayari, okay, yung degradation nung mga abnormal proteins natin, those that are misfolded or denatured, okay? So, ito yung ating target protein for degradation, okay? Adding, no, itong target protein natin na to, hindi po siya i-degrade ni 26S proteasome complex unless itag po natin siya with our ubiquitin, okay? So, kailangan po natin si ubiquitin natin, okay? So guys, no, um, the degradation pathway if you can see here requires now this polyubiquitin chain. This is your polyubiquitin chain. Okay, ito po sila, yung mga ubiquitin. Kaya siya tinawag na polyubiquitin kasi multiple ubiquitin proteins. 'Di ba? Dikit-dikit sila. So, this is now a chain. Okay? So, guys, no, in activating now, okay, your ubiquitin, we have okay, or we need these enzymes, your enzyme 1, enzyme 2, and enzyme 3. Okay, so these enzymes are necessary for us to form your polyubiquitin chain. So ito po siya. This is your polyubiquitin chain. And this polyubiquitin chain will attach now to our target protein. Okay, ibig sabihin natin, napaka-importante po ang attachment ng ating polyubiquitin chain doon sa ating target protein. Okay? So we have to tag, okay, the target protein with our polyubiquitin chain. Okay? For it, okay, to be degraded by our 26S proteasome complex. Then after tagging, no, your your polyubiquitin chain will present now this protein to the 26S proteasome complex. Okay? Adding ang kukunin lang po no 26S proteasome complex po natin ay si target protein. It will enter po dito sa lead. This is the 9S RP sa lead. Okay? Papasok po siya diyan. Okay? So, if you can see, yung ubiquitin natin or the polyubiquitin chains, okay, will be dismantled, okay, to how do you call this one? To free now, okay, yung mga ubiquitin proteins natin. These proteins will be recycled for another use, okay? So, ang tatanggapin ni 26S protein complex natin or proteasome complex natin adding, yun pong ating target protein, okay? So, the regulatory particle that forms the lead, okay, of the main protein destruction chamber will recognize the polyubiquitin tag. Okay? So, dahil meron yung polyubiquitin tag, na-recognize niya ngayon, okay, and this will lead to the entry of your target protein, okay, into the destruction chamber. Okay, so this is the destruction chamber or your 20SCP. Okay, so the regulatory particle on the opposite side of the chambers releases short peptides and amino acids after the degradation of the protein is completed. So the degradation will happen here. And then, the 19SRP or the regulatory protein naman dito sa base natin, okay, dito mare-release ngayon yung na-degrade na proteins natin. Okay, so this is the target protein. It will enter the 9SRP doon sa lead and then it will be degraded in the 20SCP or in the chamber, in this chamber. And eventually, okay, it will be released, okay, as degraded proteins, okay, or short peptides and amino acids dito po sa ating 19SRP sa base. Okay, so that's your proteasome-mediated degradation. So tatandaan ninyo in your proteasome mediated degradation, there's a need, okay, for us to tag first the target protein with your polyubiquitin chain. And to form the polyubiquitin chain, okay, we have or we need the enzymes, okay, to form this chain. Okay, so this chain now will present the target protein, okay, dun po sa ating 26S proteasome complex sa kanyang 19S RP or yung kanyang lead. Okay, and eventually it will be presented, okay, for degradation, and then the polyubiquitin chain will dismantle, okay, and then these proteins now, yung mga ubiquitin proteins na ginamit natin to form the polyubiquitin chain will be recycled for another use. Okay, so that's your proteasome degradation or mediated degradation. Okay, so we have here the malfunction of proteasome-mediated degradation. So we have the loss of proteasome function and the accelerated degradation. Okay? For the loss of proteasome function, okay, bakit nagkaroon ng loss of proteasome function? Kasi guys, nagkaroon tayo ng mutation in the system of ubiquitin activating enzyme. Di ba sabi natin yung mga enzymes na yun guys are necessary to activate your ubiquitin for them to form now your polyubiquitin chain. At ano ba yung gamit ng polyubiquitin chains natin? They're necessary in tagging now, okay, yung ating target protein. So since there's mutation in the enzymes necessary for the activation of your ubiquitin chains, then there will be problem, okay, in the degradation. Kasi kailangan natin yon, di ba? Prerequisite, okay, yung formation ng polyubiquitin chain natin. But there's problem with the enzyme, okay, that activates now the ubiquitin proteins, okay? So this will lead to what? This will lead to decreased protein degradation, okay? And this lead now to accumulation of this protein in the cell cytoplasm. So guys, no, this is an example of a problem that we can see in your Angelman syndrome and Alzheimer's disease. So I want you to research on that, okay? The relationship of the problematic proteasome mediated degradation, okay, in your Angelman syndrome and your Alzheimer's disease, okay? So for naman, so again, dito loss of proteasome function, okay? Dito naman accelerated naman, okay? Yung degradation. Okay? Bakit nagkaroon ng accelerated degradation naman? Dito, nagkaroon kasi naman tayo ng overexpressed proteins. Okay? So, proteins that are involved in the system, okay, in the 26S complex as well as maybe in the tagging, okay, of your target protein, nagkaroon ng overexpression. That lead now or these lead now to the accelerated degradation, okay, of your proteins. Okay? So a very good example we have in your infection with your HPV or your human papillomavirus. Okay? So discovery of specific proteasome inhibitors holds promise for treatment of cancer and certain viral infections. Okay? So please, no, I want you to research on how, no, your Angelman syndrome and Alzheimer's disease, okay, or how is your or how's the problem in your proteasome-mediated degradation, okay, related to your Angelman syndrome and that of your Alzheimer's disease, okay? So next we have here your rough endoplasmic reticulum, okay? So guys, no, yung rough endoplasmic reticulum po natin, it can be sorry, rough endoplasmic and then your smooth endoplasmic. Okay? So rough po siya, okay, because it has no attached ribosomes. And I know in basic biology, you know that. Okay? So when we say endoplasmic reticulum, it encloses a series of intercommunicating channels and sacs. Okay? And we call this as your cisternae. Okay? So, papasadaan ko na lang to guys, no? You can read this sa libro ninyo. Okay, I just discuss more dun sa mga pathways po natin. Okay, but for these organelles, I believe you have learned much from this or much, okay, about this, okay, in your basic biology. So please do have a review of that, okay? So this is an ATEM image, okay, of your rough endoplasmic reticulum. So if you can see here, ito yung kanyang interconnected membrane limited, okay, flattened sacs, okay, and we call this as your cisternae, okay. So they are actually studded, okay, with your ribosomes, kaya siya tinawag na rough, okay, kasi meron tayo nung smooth, okay. So guys, no, your rough endoplasmic reticulum has the following function. Memorize na memorize nyo na to, di ba? They are for synthesis and segregation of your proteins, those that are not destined for your cytosol, okay. Initial glycosylation of glycoproteins, post-translational modification of newly formed polypeptide, and of course, the assembly of multi-chain proteins. Okay? So, yeah. Please review din ito dun sa table na binigay ko sa inyo last time, di ba? The characteristic appearance of your organelles under your TEM and your SEM. Okay? Under your light microscopy, okay, we call, okay, your rough endoplasmic reticulum as the portion or the portion where we find rough endoplasmic reticulum as your ergastoplasm, okay, under your LM. Okay, so these are the protein synthesis, okay, processes. We have your transcription and translation. So these will be discussed with you sa cytogenetics ninyo. Okay? So we have here the clinical significance, okay, for your RER. So a few diseases are characterized by an inability of the RER to export a mutated protein, okay, to your Golgi. So a very good example, adding no, is your alpha-1 antitrypsin deficiency, okay? So your 1AT, okay, is a amino acid substitution, okay, that renders now the RER, or sorry, amino acid substitution, okay, or a mutation can render now your rough endoplasmic reticulum to be unable, okay, to export now your alpha-1 antitrypsin, okay. So that mutation will lead now to a decreased activity of your alpha-1 antitrypsin in the blood, okay, and of course the lungs, okay, and will actually cause now deposition, okay, of defective alpha-1 antitrypsin in the rough endoplasmic reticulum of your liver and your hepatocytes. So this will result to what? Emphysema or your COPD, an example, emphysema or COPD, and your impaired liver function. So ano ba tong sinabi ko? Okay, so guys, no, ulitin ko, when there's a mutation, okay, that causes now or that renders your RER, okay, to become incapable, okay, of exporting your alpha-1 antitrypsin, okay, that will lead to a decreased activity, okay, of your alpha-1 antitrypsin in the blood, okay, and of course in the lungs as well as in the liver of the hepatocytes. Ano ba yung kahalagahan po ng alpha-1 antitrypsin natin? Guys, no, your alpha-1 antitrypsin protects the lungs, okay, from your neutrophil elastase. Ano ba yung neutrophil elastase? Guys, your neutrophil elastase, okay, are necessary to digest now cells or bacteria in the lungs, okay, to promote healing. Okay? However, sabi nga natin, in patients with alpha antitrypsin deficiency, anong nangyari? Okay? Nagkaroon na tayo ngayon or nawala na tayo ng protection with neutrophil elastase. 'Di ba? Si neutrophil elastase, sabi natin, it will damage the cells or bacteria nung lungs natin. Okay? So as to promote healing. Okay? However, dahil nagkaroon tayo ng alpha-1 antitrypsin deficiency, wala nang magpo-protect, okay, doon sa lungs natin, lalong-lalo na dun sa normal, okay, or healthy lung cells natin from your neutrophil elastase. So instead of only damaging now the cells, okay, or only digesting the damaged cells and bacteria, nada-damage niya na rin or nada-digest niya or nasisira din nung neutrophil elastase natin, pati yung mga healthy cells sa ating lungs. Okay, that leads to the development of your emphysema, okay, or your COPD. Okay, so that's your alpha-1 antitrypsin deficiency. Okay, so yeah. Okay. Sa ribosomes ito, nailipat lang siya. Okay. So next we have your ribosomes. So I know you know that your ribosomes, okay, are the site of protein synthesis. Okay. And this will be transmitted to your Golgi finally for packaging and for the secretion, okay, out of the cell. Okay. So aha, okay. So, dito ako mag-e-end, no? For your ribosomes, tatandaan ninyo ito, guys. Yung ribosomes po nung ating mga prokaryotes or prokaryotic cells and the eukaryotic cells ay magkaiba. And these were actually exploited, okay, by researchers to discover now your antibiotics, okay, that will bind bacterial ribosomes. Bakit? Kasi nagiging specific sila. Kasi nga magkaiba daw yung ribosomes ng mga prokaryotes like your bacteria sa ribosomes ng mga eukaryotes. Okay, like human cells. Okay? So, ibig sabihin, yung mga antibiotics natin na ang target niya ay yung bacterial ribosome, nasisira niya yung bacterial ribosome, but without harming, okay, the ribosomes in the cells, okay, of the individual with an infection, okay, of that bacteria. Okay, caused by that bacteria. Okay? So several types of antibiotics inhibit protein synthesis at ang target po nila ay yung bacterial ribosomes. A very good example of these antibiotics, we have your aminoglycosides like your streptomycin, your macrolides, your lincosamides, and then your tetracyclines and chloramphenicol. Okay? So, yun yung kanilang guiding principle. Okay? The difference between the ribosomes of the prokaryotes and the eukaryotes. Okay? So, the researchers, again, exploited this knowledge to come up with medicine that will target bacterial ribosomes. Okay? Not, and that do not harm now the ribosomes, okay, of the human cell. Okay? So that's okay the idea, okay, or that's the principle, okay, they use now in developing now yung mga antibiotics natin that inhibit protein synthesis. So examples are the following, okay? Okay, so for smooth endoplasmic reticulum, smooth siya kasi it lacks now bound polyribosomes. Okay, or they lack now yung ribosomes natin. Okay, that makes the rough endoplasmic reticulum rough. Okay, so your smooth endoplasmic reticulum is actually continuous with your RER. Okay, and the cisternae are often more tubular compared to your rough endoplasmic reticulum. And they are not basophilic. Bakit? Kasi nga, they do not contain now bound ribosomes. What makes kasi your RER or rough endoplasmic reticulum basophilic are actually your attached ribosomes. Okay? So your smooth endoplasmic reticulum has the following function. We have for glycogen and lipid metabolism, detoxification, temporary calcium sequestration, and of course, phospholipid synthesis. Okay? So guys, no, please do review your book, okay, sa functions ng mga organelles na hindi ko nagaanong diniscuss, okay? Kasi these were already discussed, okay, with you sa basic biology, okay? So that's why I only focus dun sa mga pathways natin, okay, to explain it to you, okay, more. Okay? So this is now an image, okay, of your SER in your Leydig cells, okay, of the testis, 'di ba? That produces now steroid hormones, okay? So yung mga cholesterol natin, mga sterols natin, they're being produced, okay, dun sa ating smooth endoplasmic reticulum. Okay? Kaya makakita nyo madaming ganito sa mga cells that produce now steroid hormones. Okay? So finally, we have your Golgi apparatus, okay, the organelle that was named after Camilo Golgi, okay? So it's composed of smooth membranous sacules containing enzymes, okay, and the functions are the following, okay? So sabi natin, di ba siya yung LBC ng cell natin. So it functions for sorting and packaging, okay, of our proteins, okay? So this, okay, is your Golgi. So we have the cis phase and your trans phase. Okay? So kung maalala nyo siguro to, yung incoming transport vesicle is the cis phase and yung papalabas sa kanya is the trans phase. Okay? Maalala nyo yung sa endosomes natin, yung Golgi-derived secretory pathway, lumalabas yung proteins dito sa transphase. Papasok sila from the cis. Okay? So ulitin ko, transport vesicles merge with Golgi receiving region and that's the cis phase. Okay? And it's the transphase, okay, that um where no completed protein products are carried away from your Golgi. Okay? So kung matatandaan yung mga drawing kanina dun sa inyong endosomes, 'di ba? Malala nyo 'yon. So dito sila nagpi-pinch off, okay, yung proteins natin, okay? Dun sa lysosomal biogenesis pala kako sana, okay? And then your mitochondria. So I know, alam nyo naman si mitochondria, siya ang powerhouse of the cell. Okay? So, yan. This is usually in abundance, okay, pagdating natin sa mga cells like your cardiac muscle cells, yung cells of some kidney tubules. Kasi these are the cells na that require high energy for metabolism. Okay? So they have abundance of mitochondria. Okay? Yan, ito po siya. So yung mitochondria po natin adding, meron siyang outer and then inner membrane. So yung inner membrane, it's continuous. Okay? Papaloob. So kung makikita nyo, ito sila. Ayan. Pumapasok siya sa loob. 'Di ba? So that is to form now the crista, okay, of the mitochondria. Okay? That's it. So this is a time image of our mitochondria. So if you can see here, no, the series of infoldings, yung mga crista niya, ito sila, oh, yung, okay, yung mga crista or cristae. Okay? So they are actually a continuation of the inner membrane. Okay? Yan. Okay. So next, we have your peroxisomes. Your peroxisomes, guys, are spherical organelles, okay, that are enclosed by a single membrane, and they are named for the enzymes that they produce, okay, that are necessary now for the degradation of your hydrogen peroxide. Okay? So, oxidases, catalases, okay, ang meron po sa kanya. Okay? So, they have similar structures with lysosome, but they do not contain lysosomal hydrolases. Okay, that's naman your peroxisomes. Okay, so your peroxisomes, guys, forms in two ways. It's either through budding of the precursor vesicles from the endoplasmic reticulum. So, galing daw sa endoplasmic reticulum, yung precursor vesicle. O kaya naman, yung existing peroxisomes natin nagdi-divide into new peroxisomes. Okay? So, that's your peroxisome. So, your peroxisome, again, is an example pa rin po ng membrane-bound organelle. Okay? Pero siya po ay merong single membrane lang. Okay? So, yan. Ano po yung function ng peroxisome natin? It's also for lipid metabolism. Okay? So, anong ginagawa po ni peroxisome? It convert now your reactive oxygen to, how you call this one? To substances, okay? Or yung mga reactive oxygen natin like your hydrogen peroxide to safer ones like water and oxygen. Okay? So, yun po yung function ni peroxisome. Okay? So, for non-membranous organelles, guys, no, this will be your assignment. Please do read your book. Okay? Alam nyo na to. We have the microtubules, your microfilaments, okay, your intermediate filaments. Please memorize this, the 5 major classes of intermediate filaments. You will encounter this pagdating nyo sa CC ninyo. Okay, so please, even in histopath, okay, memorizing nyo lang sila. Okay, kung saan po sila nakikita. Okay, cytoplasmic inclusions. Okay, just have a review of this. Okay, nice to know. But you will have deeper discussion of this sa higher subject po ninyo. Okay? So, and for these ones, we have the appendages. You will have no a better discussion dito, lalong-lalo na sa ano, mas ano siya, tawag dito, mas essential siya pagdating nyo ng discussion naman sa bacteria ninyo. So, sa microbiology po siya. But, I'm not telling na hindi nyo po to aralin sa libro ninyo. Okay? So, please do read this. Okay? Sa libro po ninyo. Okay? So, that's all for your cell cytoplasm. Okay, so guys, no, please do review and expect a quiz. Okay, so this is a 2-week topic. Okay, so I expect na nung first week nabasa nyo na po yung presentation and yung book ninyo. Okay, so that's all and thank you very much.